everyone. Welcome for this webinar. I see that we have more than 200 participants. It's very good. I am Madalena Pimentel from the University of Lisbon, and together with Joana Azeredo from the University of Minho, both in Portugal, we are the organizers of the Virus of Microbes 2020, which will be held in Portugal. This conference is the official meeting of the International Society for Virus of Microbes. This meeting takes place every 2 years, but due to the pandemic situation that we are all living, we had to postpone this conference to July 2021. So the organizing committee, together with the support of the ISVM and the phage directory, decided to organize a series of webinars to fill this 1 year gap and to keep the communication between the phage community. So we have started this series of webinars last September, and we have each webinar every month. That started last September and will end on February 2021. And today we have now the third webinar, which is, and these webinars are under the theme of Take a Walk on the VOM side. So the today webinar, the third webinar, is focused on the virus-host interactions, overcoming the cell barriers. So on behalf of the organizing committee, we would like to thank all the speakers, the chairs, and our sponsors, and of course the audience for participating and allow us to make this possible. So very welcome to this session. So for today, we have three short talks from 3 speakers, and I will be sharing this session together with Rai Young, and it's a pleasure and an honor to share this session with him. We encourage all the participants to ask questions to the speakers at the end of the talks. And you can ask your questions on the chat and only through the chat. And at the end, we will select some questions for each speaker. So it's now my pleasure to introduce the first speaker, Martin Loessner. Martin is a professor at the Swiss Federal Institute of Technology in Zurich. He is the head of the food microbiology group in the Institute of Food, Nutrition and Health of the Department of Health Science and Technology. His research has been focused on the study of food pathogen and the relation with their bacteriophages, particularly the Listeria monocytogenes. And today he's going to talk about how phage-induced modifications of the cell components is related to attenuated virulence of bacteria. So the title of his talk is Sweet and Sour, How Cell Surface Glycosylation Affects Phage Sensitivity and Virulence. So thank you, Martin, and the screen is yours.
Thank you very much, Madalena, and good morning to everyone, good day, good evening, wherever you are. We have only 10 minutes and need to keep going. So I would like to introduce you into a topic that I call Sweet and Sour, how cell surface glycosylation can affect phage sensitivity and virulence. And what we see here is just a basic overview of some of the virulence-associated phage receptors on bacterial cells. We see that on gram-positive bacteria, there may be flagella, there may be some carbohydrates, proteins, the same on gram-negative bacteria. However, for today, we will just be focusing on one molecule, more or less, which is the wall-teichoic acids. Now, wall-teichoic acids are major determinants of antigenicity in gram-positive bacteria. What we see here is a Listeria cell, and this is about how the wall of this Listeria cell looks like. What's a bit misleading here is that teichoic acids are actually underrepresented. They normally make up about 70% of the dry white of such a cell. And there's 2 types of them. on Listeria cells, there's a type 1 wall teichoic acid here, and it's a polyribitol phosphate, which is then substituted with a number of carbohydrates, such as these sugars here, glucose or N-acetylglucosamine or rhamnose. And because this is an acid and this is the sugar, I call it sweet and sour. And actually this corresponds quite nicely to the very talented scientists that is leading these studies, Dr. Yang Shen from my group, and his diet is also favoring sweet and sour, as you can imagine. Yang is a wonderful guy, and I'm happy to have him here. He is responsible for most of these data. Now, what I'm going to be showing you now is how can we actually determine the sugar composition? Well, we can use various physical methods like NMR for structure determination or mass spec for analysis of these sugars. A far easier method, however, is to use phage components again. What you see here is a fluorescently labeled phage protein, an endolysin cell wall binding domain, which can recognize the terminal N-acetylglucosamine on those polyribitol phosphates. And it labels them nicely in green. If that is gone, no label. So that's a very easy method. We've actually published quite a bit of papers on this, and there's one very recently that was published which shows you a complete toolbox of these proteins to determine the full glycosylation pattern. Now, what we also found is that phage affects these glycosylation patterns. What we see here on the left side is the full-blown glycosylated version, which carries both rhamnose and N-acetylglucosamine. It's one of the dominant Listeria serovars. It's called 1,2, not 1/2, but 1,2. If the N-acetylglucosamine is not there, it becomes a serovar 1,2 star. If the rhamnose is missing, it becomes a serovar 3, and if both are lacking, it becomes a serovar 7. Turns out that these serovars are just point mutations, and these point mutations occur in genes which are responsible for the glycosylation, and phage selects for those. And it also turns out that these guys are losing their virulence. They're actually becoming avirulent, almost harmless bacteria. So phage does something good here. And we go a bit further. We go to the next type of wall teichoic acid, which is the type 2 wall teichoic acid. And you see there's more variation here. And they're a bit different built because the chain continues not on the other side of the ribose C5 here, but it contains from the N-acetylglucosamine, which is an integrated component. And there can be many substitutions on here. Again, sugars such as rhamnose, glucose, and galactose. And galactose is a key component here. And I would like to focus a little bit on that one. And that has been the subject of a PhD student that more or less recently graduated from this lab here, Eric Summerall. And what he found is that when a serovar 4B, which is the most virulent Listeria serovar, if you challenge this by phage, you end up with a version of bacteria that it has become phage resistant, but it also lacks the galactose. And this is a serovar conversion from 4B to 4D by phage resistance. And we have been looking into this a bit further. We've also looking into the biochemistry, into the building of these molecules by the Listeria cells. And there's a bunch of enzymes that are located in the membrane. These are these enzymes here, and they're responsible for the wall teichoic acids. And there's another enzyme over here that is responsible for galactosylation of the lipoteichoic acids. However, we will focus on these here. And now what Eric has been doing, he has been creating mutants with single knockouts in these genes. And guess what? What he found is that galactosylation of wall-TA, but not of lipoteichoic acid, mediates phage absorption of different phages, various phages, such as this one here. You can see if we create knockouts in this one single gene, you completely lose absorption of this phage or of this other phage. But not only that you lose phage absorption by knocking out these genes, you also lose virulence. The loss of the wall-TA galactosylation, again here, over here, on the left-hand side, you see what happens to the major virulence factor internalin B on the surface of the bacteria. it drops by almost 2 logs. So 99% of the internal NB is lost from the cell surface. This has massive consequences for invasion, for instance, of HeLa cells. You see here, you don't see anything here because there is no invasion anymore. Also here, that's another mutant strain that lacks the galactose. And we can then look in the internal organs of an animal model, and what we see that in the liver of these poor mice here, there is far less Listeria that features this mutation. So altogether, it is a single point mutation or a complete in-frame removal, doesn't make a difference. Phage-resistant Listeria lose their virulence. And altogether, we conclude that this is a massive trade-off between phage resistance and virulence. What we see on the left side is the full-blown normal Listeria monocytogenes serovar4b, which causes like 80% of all epidemics and outbreaks worldwide. It has the galactosylated teichoic acid, it has the internal NB, and it can cause disease by invading cells. However, if phage comes and selects for the galactose-less mutants, there's non-galactosylated teichoic acid, internal NB is lost from the surface, and these guys can no longer invade. So take-home message from this is, even if you have phage-resistant Listeria, for instance, in foods, after this food has been treated with phage to remove the Listeria, those phage-resistant bugs don't pose a real threat because they become mostly avirulent and even apathogenic. Now, the last couple slides, I want to tell you something, what we can do to circumvent this problem. Why become Listeria phage resistant? Because they want to avoid the predation by the phage. However, if we still want to kill them, for instance, in food, we can reprogram the host range. And if you want to do this, you have to look at the adsorption mechanisms. And of course, everyone knows this is the tail fibers of phages that do this. And we recently had the cover page of this journal here, where a PhD student of mine contributed a nice drawing, which more or less tells the story of what we have been doing. We just have been putting various different tail fibers on those phages. And this is the work of Matt Dunn and Sam Kilcher from my lab. And what they have been doing, they have been looking at the tail fibers in the most detail possible. They have started out using X-ray crystallography to study their structure, their makeup. Then they have been putting them together from scratch, from the basic components by rational engineering. And what we have been ending up with is the wild-type phage can only infect one given type of Listeria cells. And imagine this becomes resistant by losing the galactose, then the phage can no longer do anything. However, we can go through a series of mutations and rational design and come to a designer phage. And that designer phage, actually, I'm going to show you what it does now. The wild-type phage can only infect one single type of Listeria cells, which is the 4B. The designer phage, which features a newly made up rational design tail fiber, can infect all these different Listeria strains. So that would be the one that you want to be using to circumvent the loss of a single sugar for phage insensitivity. So, and that brings me to the end of my talk. There is a bunch of people, of course, that work in my lab, and I would like to thank all of these, and I would like to thank you for your attention. I'm happy to take any questions that you may have. Thank you.
Thank you, Martin. So I encourage all the participants now to write your questions in the chat. And please introduce now the second speaker.
Okay, I hope I don't screw this up. I'm the oldest person here and I can claim technological incompetence very easily. That addresses part of the issue. I've known Carlos, our next speaker, Carlos São-José, since he was a graduate student and actually was in his PhD defense back in the early part of the 21st century. Carlos has always had a penchant for understanding not only the role of the bacteriolytic enzymes in degrading the cell wall, but also in how they're controlled. And in fact, his work and his colleagues in Mario Santos's lab back in the early 2000s really shattered the paradigm and was the first first papers that showed that there were multiple different ways that lysis could be controlled. His talk today is License to Lyse, Factors Impacting Endolysin Lytic Action and Gram-Positive Bacteria. So Carlos?
Okay. Okay. So thank you, Rai, for the introduction. So in this talk, I will briefly present the work we are doing with phage endolysins targeting gram-positive bacteria, focusing on factors impacting endolysin lytic action. And we think this is important because endolysins are being explored as potential alternatives to fight drug-resistant bacteria. So endolysins are cell wall degrading enzymes, and they are essential to cause cell lysis at the end of phage infection for virion progeny release. Those endolysins for gram-positive bacteria have a typical modular organization carrying one or two catalytic domains in the N-terminus responsible for peptidoglycan cleavage, and a cell wall binding domain at the C-terminus carrying one or more cell wall binding motifs. And of course, these endolysins have the potential to cause lysis when added from the outside, and this sets the basis for their exploration as antibiotics. Now, one thing we observed for several endolysins is that conditions promoting dissipation of cells membrane potential, the PMF, can greatly enhance bacterial susceptibility to the lytic action of endolysins. And this is well illustrated here for these three endolysins added to target cells in rich culture media. At these concentrations, you can see that there's no lysis or very poor lysis, here observed by these gray optical density curves. But in presence of agents that dissipate the PMF, like for example, niacin and gramicidin, you can see that lysis is very fast immediately after addition of the endolysins. And we think this reflects the natural context of action of endolysins. Most phages follow the so-called canonical lysis model, meaning that endolysins are translocated to the cell wall through these holes made in the membrane by the phage holins. And of course, these holes also cause PMF dissipation, and we showed that this can be a requirement for full sensitization of cells to the lytic action of endolysins. And in fact, this was first shown for endolysins that are exported to the cell wall through host cell export machinery, like for instance, the SEC pathway. And so based on these observations that PMF collapse can potentiate the lytic activity of endolysins, we search for agents that could mimic the olin action in hypothetical therapeutic contexts. And of course, the best choice were antimicrobial peptides, because most of these agents act precisely by promoting PMF dissipation. And so the question was, could AMPs be used to promote or to stimulate endolysin lytic activity? And the answer is yes, at least in vitro for several endolysins. Here I show the example for the endolysin of Staphylococcus phage Phe11, added here to TSB medium. You can see that at this concentration, there's no lysis, there's a stable optical density curve, but in presence of an AMP, you see that endolysin is quite fast and massively after endolysin addition, in contrast to the AMP alone that doesn't cause any lysis. When these two agents are acting together, we see a 3 log reduction on cell viability compared to roughly 1.5 log reduction in presence of the AMP. And of course, as expected for the lysis curve, the endolysin by itself has barely any impact on cell viability. And quite remarkably, in presence of the AMP, we could reduce by 10-fold the endolysin concentration, and we still see this lysis with almost the same cell death. So the question is, what's causing this tolerance to endolysins when bacteria are happily growing in complex media? And why is this tolerance eliminated when in presence of AMPs or other PMF dissipating agents? And in fact, several studies have implicated wall-taker acids in shielding the peptidoglycan to phage endolysins and other lytic enzymes. And when we inhibit wall-taker acids with tunicamycins, we see that the endolysin can easily lyse cell cultures, and this correlates with an increase of about tenfold to cells carrying no wall-taker acids, as you can see here, by using this endolysin GFP fusion. And so we tested if the AMPs were promoting endolysin binding to cells, and it does, as you can see here, by using these two protein concentrations. But at the same, so here we have about 2 to 3 fold increase in the endolysin binding in presence of the AMP. But as you can see also here in this condition where we don't have any AMP, the protein bound to cells is slightly higher than condition in presence of AMP. And as I showed you before, here in this condition, which corresponds to this curve here, we don't have lysis, whereas in this condition, corresponding to this lysis curve, we have clearly cell lysis. So yes, the AMPs are promoting endolysin binding to cells, but this by itself cannot explain the lysis phenotype we observed in presence of AMPs. And so these agents must be stimulating lytic activity by other means. And we have some ideas to test what eventually we can discuss later. Okay, another factor that we are interested is on endolysin domain multimeterization and how this affects lytic activity. So most endolysins targeting gram-positive bacteria are described as monomeric, and this is the most common domain configurations. But there are also some multimeric endolysins, and the first described was ply-C, which is composed of ply-C-A and ply-C-B subunits, so the catalytic domain and the cell wall binding domain. The holoenzyme is composed of one ply-C-A subunit coupled to a ring of 8 cell wall binding domain subunits. And in 2015, we described another multimeric endolysin, but in this case, the two subunits were encoded by a single gene, thanks to the presence of an internal in-frame translation start site, a TSS, located right here, immediately before the cell wall binding domain. And this site produced or drive the independent production of this cell wall binding domain. And based on our biochemical analysis, we have proposed an oligomeric state composed of a single domain corresponding to the full-length endolysin coupled to 3 cell wall binding domains. These are other endolysins with known TSS. Some of them were meanwhile confirmed multimeric, and the 3D structure of this one was recently solved, and the authors found the 1, 2, 3 oligomeric state that we have proposed earlier in our work, as you can see here in this 3D structure. So we became interested in understanding or investigating how common are these endolysins with TSS and if they contribute to endolysin multimerization. And so we started this work very recently. We retrieved the endolysin genes from the phage lytic proteins database. We predicted TSS sites using this tool, RBS calculator, in the +1 frame, and we ranked the TSS sites according to their translation efficient rates. And we found several endolysins with putative TSS targeting different gram-positive bacteria, as you can see here, and also microbacteria. And some of these genera are enriched in endolysins with high score TSS, like for example, Streptococcus, Lactococcus, and Clostridium. And here I show for reference the endolysins with known TSS. And so here you have some examples of endolysins with high score TSS. As you can see, there's high diversity in terms of catalytic domains. And most of the TSS, of the putative TSS, are located here at the C-terminus of the endolysins, so close to the region encoding the putative cellular binding domain. But there are also some examples of TSS located upstream the catalytic domains, like here and here. And so we just had the time to clone and express one of these endolysins. In this case, an endolysins from a Streptococcus pyogenes phage. And as you can see, we obtain the two expected proteins, the two expected polypeptides. So a polypeptide corresponding to the full-length enzyme and a C-terminal fragment corresponding to the C-terminal, to the product starting here at this TSS. And so, of course, now we are starting the studies to see, to try to understand how these two polypeptides contribute to lytic activity, and of course, if they associate in a complex. And so just to finish, I would like to thank my team, so Raquel, Daniela, and Ana Gouveia, and of course also IMED for all the support, and FCT for funding. Thank you very much.
Okay, Carlos, thank you for your wonderful talk too. We'll start with questions for the first speaker, which is Martin. Martin, I have several questions here for you. Let me try to select some. One of the questions is, from an anthropocentric point of view, do you think changes on galactosylation is a side effect or something actually beneficial for the phages infecting Listeria monocytogenes?
Hi, Madalena. I'm actually prepared already for answering these questions. I'm sorry. I just try to be as efficient as possible. And I printed all of them out and marked a few which I'm ready to answer. By the way, this question is hard to answer. I didn't mark that one because I don't really know what it's supposed to mean by the anthropocentric point of view, but the galactosation, the loss of galactose brings about massive consequences for the bacteria. And this trade-off for becoming phage insensitive is so massive that in nature, we find them very, very rarely. They almost never are found, these super seldomly occurring serovars. And there must be some massive pressure, selective pressure on keeping all these sugar molecules on both types of teichoic acids. All right, I prepared a few or marked a few here. Do we see the same resistance develop in other bacteria such as enterotoxigenic E. coli? Similar questions were also asked by others. That's a very good question, and actually exactly the same thing. So it's not loss of galactose from teichoic acids, it's loss of sugars, for instance, from the LPS side chains. It's shortening of the side chains. You become a rough mutant, for instance, of E. coli or Salmonella as a result of phage challenge. So it actually does occur in the gram-negs as well. Next question, how long does this resistance to the phages?
Can I just put some questions there about gram-negative bacteria? Because you also have modifications in the peptidoglycan and sugar modifications. So that could be also a target in gram-negative bacteria?
Well, gram-negative bacteria have, for the most of them, the same kind of peptidoglycan. The only variation they have is in the decoration of the outer membrane. So the sugar side chains are highly variable, but the peptidoglycan itself is highly conserved. So all the changes that happen in the gram-negs actually happen to the sugar side chains, and also the gram-positive, same thing. There's mostly changes to the carbohydrates and not to the cell wall. There's only very few examples, for instance, in Staph, where the pentaglycine bridge in Staph aureus may be modified in response to enzymes that can cut it, but that's about the only exception there. So how long does this resistance to the phages last? A super interesting question. And since it's a mutation, it lasts. We have not seen any reversion in terms of going back to the phenotype of a fully glycosylated variant, at least not in the time frames that we used in the lab to look for this. Do you still see resistance to the designer phage? Also a very cool question. We actually haven't tested this in all detail, but what we have seen is that there is very, very little survivors, right? Very little resistance. And that is because these phages were modified a little further than I have been telling you. We have also removed their lysogeny control regions so they can no longer integrate. What we haven't been doing, however, is taking care of CRISPR motifs. So Listeria does have CRISPR systems, and it happens that some of these phages are actually been interfered by a functional CRISPR-Cas system in some Listeria strains. So that is another possibility to interfere with phage infection.
Okay, so
I have
one more question, okay?
Yeah.
Only
one more?
Yeah.
Oh, okay, one more. Let me see.
Move to the next speakers.
Yeah, here's an interesting one. That comes from Joanna, actually. I like this question. Listeria phage resistant becomes less virulent, but how about their persistence in the food industry? How about their ability to adhere and form biofilms? This has actually been looked at and also published. And those mutants which lose these components, especially glucnac and acetylglucosamine from the surface, they become less capable of adhering and forming biofilms. So it's another benefit for using phage in the food industry, if you want, that the phage-resistant mutants not only become less virulent, they're also less persistent in the production environment. Thank you.
Okay, thank you so much, Martin. I recommend that if some participants still have any questions may contact you directly. And please, Rai, let's move to the next questions to the speakers 2 and 3. Can you please?
Yeah, so first of all, it's really nice to see that our president's predilection for only answering questions that he wants has been replicated here. Thank you, Martin. It was great stuff, though. So, Carlos, I've had several questions here about the AMPs. First of all, which AMP did you use?
Okay, so that's a derivative of so it's a low toxicity version of an AMP called SMAP29, so sheep myeloid antimicrobial peptide.
Okay, so the question, 2 people asked the question, are these...
But it works with other AMPs also. We tested with other AMPs.
Well, that answers the question, yeah. Really.
Okay.
And one question was, do you think license could actually increase the access of AMPs to the membrane?
Theoretically, yes, but in fact, when endolysins start to break the cell wall peptidoglycan, at least in gram-positive bacteria, they will immediately lyse. So, I don't think it could be that useful.
Okay. One question from Dan Nelson about the alternate start codons for that part of the talk. Are the alternate start codons of the same ilk as the primary start codon for the full-length gene, or they tend to be more the rare starts like TTG?
Well, I don't have numbers, but we found alternative start codons, so GTG and TTG. And in fact, one of, if I remember well, one of those clostridial endolysins that have the same phenomenon and are also multimeric, I think the smaller polypeptide is produced from a GTG start codon.
So as I'm sure you know, we've shown, lots of people have shown that there's been regulation of the translational starts at the starts of Poland. Is there such a thing going on, or is it a constant ratio just based on the strength of the
we don't know about that. We have ranked this TSS with that tool that I mentioned, the RBS calculator, and it takes into account the host cell. So it's so in principle, those TSS with the high score are likely to work at least in the host. Of course, when we will express in E. coli, we don't know what will happen, but at least for this one that we have tried, the production seems to be very efficient of both polypeptides.
So, Madalena, I'm sure you're going to give me a ding dong when I'm supposed to shift to speaker 3. But Carlos, one thing you didn't address, and I guess it's because you want to save it for later. But what do you think is actually going on now? If it in the physics of activation of these things, is there something special about the compartment of the gram-positive peptidoglycan that makes these proteins sensitive to the energized state of the cell membrane?
Well, there are some studies that raise the possibility that the PMF can act directly in repressing endolysin activity, because so the PMF is composed of two major components, so the electric potential and the pH potential, the pH gradient. And and this implicates also wall-taker acids, because it was shown that they contribute to protonation of the cell wall environment at least close to the cytoplasmic membrane. And so we can imagine that there's some an acidic environment close to in the cell wall environment that can it may happen that it restrains endolysin activity. And so when we dissipate the delta pH, that could be a reason for endolysin activation. But we are testing other possibilities. We actually, at the moment, we don't know if the antimicrobial peptides are somehow allowing the endolysin to access to other sites in the cell wall that otherwise are shielded by the wall-taker acids. And those sites may be crucial to maintain the cell wall integrity. And of course, we want also to see if cell wall composition has some impact in this activity of the antimicrobial peptides. And so we will test a battery of mutants which affect, for instance, wall-taker acid substitution, like glucosylation, allylination. And also we want to test if it happens that the autolytic machinery is activated after treatment. And this has been reported also. And so we need to check this also by making the same experiments in using bacteria devoid of the autolytic machinery, of the major autolysins. And just to mention a few examples.
Whoa, this was a great session. I think that you agree. So it's now up to me and on behalf of the organizers to close this session. And I would like to thank again our speakers for the sweet and exciting talks, and that they were absolutely well guided by our chairs. And of course, I would like to thank the audience. Today we had 318 participants, so that's really good. And if you wish to watch again this session, we have a YouTube channel. You will be seeing that in a slide that will be shown to you. Yeah, that's where you have the address of our channel, so you can see again this session, and you can also watch the previous ones. So before closing, I also would like to call your attention to the ISPM website, where you can still become a member of the society and help supporting research in virus of microbes. So that's all for today, but we will be back, and we'll be back next month for another exciting webinar, the 4th iVOM, this time on virus-host interaction, but on molecular mechanisms. And I'm very pleased to announce that we will have as speakers Rob Lavin, Rotem Sorek, and Peter Finren. So it's going to be great again. So for now, goodbye for today, and I hope to see you in December. And really hoping, and it's hoping with all my heart, to see you in person at our greatest Virus of Microbes conference next summer in Portugal. So goodbye to you all, and take care.
Goodbye.